And let me try to share my screen. And sorry about the background. The background didn't work on my computer, and even the IT couldn't help me. So you are seeing my beautiful background of my office with pictures of ALS patients and my diseased family giving me support all the time. And so I will be talking about, as Bob said, the upper motor neurons in ALS and why it is important that we improve their health. And, before I go into my talk, I want to thank the people in the lab. I want to give the acknowledgements up front. And today I will be talking about the work done by Dr. Gautam, Dr. Gench, and we have a very dynamic lab, and I am very thankful to have all our lab members and working very hard to understand the biology and pathology of upper motor neurons. And also our collaborators worldwide. But today, particularly, I will be talking about the drug discovery study that we do in collaboration with Dr. Silverman here at Northwestern. And I'm very thankful to all the collaborators that we have all around the world, and it makes the world as our playground for science. And of course, thanking all the foundations and NIH for supporting our research. And we were lucky to have received support from the first board of Les Turner ALS Foundation early in the day, starting to establish my lab here at Northwestern. We received funding from NIH and recently Spastic Paraplegia Foundation and AlongSwim. And I'm thankful to Brain Research Foundation and ALS Association for supporting the upper motor neuron research. So why do we focus on upper motor neurons? Why do we think that they are important? We think that the upper motor neurons, and we call them the corticospinal motor neurons, and they are bed cells in patients, are the spokesperson of the cerebral cortex when it comes to motor function. And what does it mean? It means that they collect information from many different regions and neurons of the brain, and they collect that information, they integrate that information, translate it and transmit it to spinal cord targets so that the voluntary movement can be initiated and can be modulated. So the movement starts in the brain. And for diseases such as ALS, in which motor neuron becomes dysfunctional and voluntary movement becomes impaired, the brain is actually is very important because that is the site where the movement is initiated. Therefore, without understanding the biology and pathology of the upper motor neurons, we don't believe that there would be effective and long-term treatment strategies for diseases of the motor neuron circuitry, such as ALS, HSP, and PLS. So the apical dendrites are extremely important because this is where the site of cortical connectivity occurs. And the axon is also very important because this is the site where the information is transmitted to spinal motor neurons. And people tell us, oh, upper motor neurons are very minimal in numbers, they are very hard to distinguish, it's very hard to study them. But we don't think the importance is related to numbers. So even as Thomas Jefferson said, one man with courage is majority. So they are actually having an important role for the initiation and modulation of movements. And that's why we have to understand why they become diseased, why they become vulnerable, and what can we do to improve their health. And again, they are clinically important because they are involved in numerous motor neuron diseases, not only in ALS, but also HSP and PLS. And their degeneration is also very early in the disease. For example, the cortical hyperexcitability or hyperexcitation occurs about 6 months prior to disease onset, and there are some groups who actually use this as an early diagnostic marker. And the cortical neurons begin to give a red flag much earlier than symptom onset. And when you actually remove SOD1 from the motor cortex of the rat models of ALS, you not only improve the health of the spinal motor neurons, but also the the neuromuscular junction and the overall health and integrity of the motor neuron circuitry, further suggesting the importance of upper motor neurons and that their early contribution to disease pathology. And now there's also new evidence that when you delete when you cross the SOD1 mice with FESF null mice, or so that you deplete all corticospinal motor neurons, you actually have a pathology that would suggest that the ALS begins mostly in the cortical neurons. And that's very important for the upper motor neuron field, that it is extremely important now that we build an understanding for their improved survival. So that's why we think that upper motor neurons must be included in drug discovery. So it is very unfortunate that today not even one preclinical assay is valid or is tested for the compounds that are in clinical trials now. None of the compounds in clinical trials have ever been tested for their ability to improve the health of the upper motor neurons. So we assume if they improve the health of the spinal motor neurons, the upper motor neurons will also be improved. But that may not be correct, because the upper motor neurons and lower motor neurons, even though we call them the motor neurons, they are totally different neuron populations. And for diseases, complex diseases such as ALS, we really have to understand if upper motor neurons also improve with compound treatment. And that's why we try to focus our attention to understand the pathologies and the solutions that would improve the health of these neuron populations. There is also a very important development in the field, and that the notion that one compound is not going to cure all patients within a disease, but some subpopulations within the disease. For example, focusing on mechanisms such as, for example, mitochondrial health, right? And patients in ALS or Parkinson's or Alzheimer's patients, some of those patients may actually develop the disease due to problems in their mitochondria. So if we focus our attention to the mechanism and try to understand the solutions based on a mechanism-based approach, there may be broad implications not only for one disease, but for numerous diseases. And as you know, nowadays also there is expanded access suggesting that compounds which pass phase 1 and phase 2 successfully may actually be considered for some of the subpopulations of diseases, so that a mechanism-based approach can be used. So the picture of drug discovery, or the landscape of drug discovery, is changing as we speak. And I think it is more and more evident that we need to understand the cellular biology, we need to understand the mechanism, and we need to begin to develop a personalized medicine approaches. So that's why it becomes even more important that we focus our attention to the diseased neurons. And translation could be possible, and I'm going to give you some examples at a cellular level. Because we've been comparing mice with human, and survival in mice is equal to survival in human, and that didn't hold true. So maybe we should really focus our attention to the cells. And of course, for drug discovery, we really need quantitative outcome measures, because how are we going to know that this patient is improving, the conditions are improving, and also the cellular outcome or the cellular health or neuron health is improving? And again, all the preclinical studies, they need to translate to patients. And how do we do that? So we believe that understanding the biology of upper motor neurons paves the way for effective treatment strategies in ALS, HSP, and PLS. And but you know, again, as Evangelos was talking about the model systems. So which models are we going to use? Which models display upper motor neuron loss? And which models are dependable, and which ones are translates to human biology? So first, initially, we started working with the SOD1 model, because that was one of the most prominent model, studied in the field of ALS. And we showed that in the SOD1 G93A mice, the corticospinal motor neurons indeed displayed an early and progressive degeneration. The same was true with the ALS knockout mice, and this was generated with Dr. Siddiq and Dr. Gang here at Northwestern. And we showed that in the ALS knockout mice, the corticospinal motor neurons also display progressive degeneration at a cellular level. The same is true for the profilin transgenic mice, that they have progressive degeneration. And most importantly, in the mouse models for TDP pathology, we used the Balo mice, there was early and profound progressive upper motor neuron degeneration. So now, here I have shown you that we have actually published work from 4 different mouse models, which are generated by mutations detected in ALS patients, and looked at a cellular level. And at a cellular level, even though, for example, these mouse do not behave like human, at a species level, they are different. But if you look at their upper motor neurons at a cellular level, you can see profound abnormalities and progressive degeneration. And again, so then we suggested that we may actually compare the upper motor neuron with the upper motor neuron at a cellular level, right? And one of the advantages of using mouse models is that there is really not much heterogeneity, and that the one pup is equal to the other pup and the one generation is equal to the other generation. You don't have the heterogeneity of the patient population. And it is clinically, it is genetically very well characterized, so that the mutation that you make or the pathologies that you have is very distinct, and it is it's very clear. So we've asked whether the problems that we see in the upper motor neurons in the mouse and upper motor neurons in human would be similar. And lo and behold, yes, we indeed find that the problems that we see, for example, apical degeneration, and I'm going to get to that, in the mouse, in different mouse models, is also observed in a vast majority of sporadic ALS and familial ALS patients, that the apical dendrites begin to degenerate. Likewise, there is mitochondrial dysfunction, very severe and very prominent. ER, disintegration of ER, problems with ER, that's also common between the upper motor neurons in different mouse models and upper motor neurons observed in sporadic and familial ALS patients. So then this brings the idea to us that if you compare cells with cells, neurons with neurons, and upper motor neurons with upper motor neurons, there's also very clear translation at a cellular level. And we actually published one of our findings about the apical dendrites recently, and I'm going to come back a little bit to that too. So these problems, the mitochondria and ER problems, were mainly observed in, with respect to TDP pathology, and they were present both in the mouse models of TDP and ALS patients with TDP pathology. So of course, this brings us the idea that moving forward in clinical trials, we really have to learn from motor neurons. So the motor neurons may actually speak to us and say, you know, this is the compound I like, or this is the compound I don't like, or I like this one better than this one, or, you know, we may have a very clear understanding of how upper motor neurons, diseased upper motor neurons, behave with respect to compound or drug treatment. So this was, again, a collaboration with Dr. Silverman, and Dr. Gainch and Dr. Gautam worked or is still working on the study, and I'm going to talk a little bit about this now. So I told you that upper motor neurons are important. Okay, fine. But how do we study upper motor neurons, right? We don't, there are very few, and every neuron, it's hard to identify them in the motor cortex. So we actually made the reporter line in which upper motor neurons are GFP. So this is published, so I'm going to go very fast. But in this case, the GFP is sustained even when the mouse is 2.5 years old, and it's in layer 5. It's in these pyramidal neurons in the motor cortex, and we, you know, confirmed with multiple studies that yes, these GFP neurons in the motor cortex are indeed corticospinal motor neurons. We published this work, so I didn't go in detail. But now, you remember I told you that we have seen upper motor neuron degeneration in numerous mouse models, SOD1, TDP, alsin, profilin, and now thanks to my colleagues, Dr. Bas and, oh my God, I forgot his name. I'll remember, I'll tell you in a minute. Gerardo Morfini. We now have the spastin mice. So we will add this to our study here to understand the upper motor neuron loss with respect to diseases of ALS and upper motor neurons. So when you cross the UCHL1 mice, the upper motor neuron reporter line, with mouse models that display progressive upper motor neuron loss, now you can generate reporter lines of disease CSMN. This is very important because even though the upper motor neurons are GFP in each of these mouse, they become diseased due to a different underlying cause, okay? So now, this is also important for a drug discovery study, because in an in vivo model, like you have the upper motor neurons GFP and you can give them the compound of interest. And here we show that in the TDP mouse, there's actually loss of corticospinal motor neurons in the cortex. But now you can see with drug treatment, can you improve that? And importantly, in vitro, which means in tissue culture, in culture, the upper motor neurons also retain their identity in culture. So you may imagine how important this is, because you will have dissociated cortical neurons in which the upper motor neurons will be fluorescent. So when you look under the microscope in the plate, the upper motor neurons will be waving a hand to you. Here I am, here I am. So when you add a compound, you just watch how upper motor neurons behave. If they are happy, they will send a very long axon, they will branch, arborize, they will be very happy. If they don't like it, they will shrink and they will die. So it is a perfect readout that you will know if the diseased upper motor neurons will like the treatment or not. So interestingly, because we have upper motor neurons that become diseased due to mutant SOD1, TDP pathology, profilin, alsin, spastin, many different underlying causes, it is also possible that one compound will be actually very much liked by one diseased neuron, for example, SOD1, but TDP will say, no, I don't like this, or TDP will like it, or SOD or profilin will not. And because there is profilin, for example, or spastin, they're important for the actin cytoskeletal dynamics. They're important for axon transport, maintaining cytoarchitectural dynamics. But SOD1 is mainly in protein aggregation, or ER stress, mitochondrial dysfunction. Similarly, TDP has problems with mitochondria ER, but also nucleus to cytoplasm defects. Alsin has problems with Golgi. It's post-translational modification, lipid biosynthesis. And every disease underlying cause is a bit different, and they have some common aspects and there are some unique aspects. So that's why when you add the compound, you may begin to tell, oh, this compound may actually be good for patients who develop the disease due to mitochondrial problem, but not so much for the post-translational modification and lipid dynamics. Or this compound is perfect for the patients who have cytoarchitectural problems, but not so much for the nucleus to cytoplasm transport defects. So we begin to distinguish even patients based on the underlying cause, so that, we can identify the subpopulation of patients. And this is extremely important for building effective clinical trials, because the heterogeneity in clinical trials is actually in part responsible for the failures that we have, especially in phase 3. If we were to include patients in a very in a way that we know that this patient would benefit from this drug, but not so much from the other one, we can improve the outcome of clinical trials. And the other beauty of having upper motor neurons GFP, a diseased upper motor neurons GFP for in vitro clinical trial, is that you don't have the patient heterogeneity. It's very clear, and there is really very little room for variation from, let's say, patient to patient, right? From culture to culture, from passage to passage. You don't have those. It is very clear, and it's only 3 days, and you get a readout. And this is reproducible, and I think this is the way to go for looking for compounds for their ability to improve the health of diseased upper motor neurons. And again, those studies would help us identify the underlying causes and the treatment strategies for subset of patients. But again, this would be important not for ALS patients, but for many other diseases in which similar mechanisms are altered. And again, the mechanisms are very complex. You have mitochondrial dysfunction, axon transport defects. You have maybe more than 16 different cellular events, canonical pathways have been attributed to ALS pathology. And that's why the heterogeneity in the patient population is huge. And we may therefore need well-defined cellular models which develop the disease based on a known factor. So as I told you, the mitochondria in ER are pathologies observed both in human and mouse. Now I'm going to tell you that the compound generated by Dr. Silverman, and he has published about these the compounds before, and we call it NU9, and we tried to see whether we can improve the health of diseased mitochondria and ER as observed in the SOD1 mice. And lo and behold, yes, we do. By improving the, and by treating mice with NU9, oral gavage, we can improve the health of mitochondria and we can improve the health of ER. So now, does this really translate to improved health at a cellular level? And at a, if you look at the misfolded SOD1, they are reduced. And if you look at the cell numbers, they come almost to the wild type levels that you rescue the upper motor neuron loss in the SOD1 mice. And I don't think any other study was able to do this, that we can actually rescue the ongoing upper motor neuron loss. And if you look at the apical dendrites, and as I told you, this is an important site for neuronal connectivity, you see them coming back to almost wild type levels, that the apical dendrites become healthy. And this is very important for the cortical connectivity, as I told you here. And again, you know, the apical dendrite degeneration in ALS patients is very prominent, and being able to restore that would be extremely helpful. So SOD1 and TDP are different pathologies, right? And we next asked whether we can actually show this in the TDP pathology as well. And even though the compounds were identified based on SOD1 mutation or SOD1, misfolded SOD1, now we actually show that even in the context of TDP, where mitochondrial problems and ER problems are prominent, as we have shown in our previous publication, again, here I'm going to show you same here, that giving the compound improves both the health of the mitochondria and the ER. And this also translates, and very much better than the SOD1, I would say, to the degeneration of upper motor neurons, that the apical dendrites become healthier and the progressive degeneration is actually eliminated. So we think this is an important achievement because we can eliminate the ongoing upper motor neuron loss at a cellular level, both within the context of SOD1 and TDP, and we can do this by focusing our attention to the cells and to the underlying cellular pathology, which we focus to the ER and mitochondria. And the keynote speaker is actually going to talk about a compound that improves the health of ER and mitochondria, and they have effects on ALS, right? But unfortunately, they haven't looked in the upper motor neurons. And here with Silverman compound, we actually see that by improving these, we improve the health of the upper motor neurons. So I would imagine that the Emilix compound would have similar effects, but of course needs to be tested for the upper motor neurons that are diseased due to different pathologies. So in summary, I think upper motor neurons are important, and I think we should all agree that they are important, especially within the context of ALS and other motor neuron diseases. Their degeneration is an early event, and they need to be included in preclinical drug development assays if we want to have effective and long-term treatment strategies. There's translation at a cellular level. So if we focus our attention to the cells, there's actually translation from what happens in the bed cells, what happens to the upper motor neurons. A mechanism-based approach will be very effective. And for effective and long-term treatment strategies, we have to improve the health of upper motor neurons. And with this, I want to thank you, and I'm ready to take any questions. Thank you so much.
Thank you, Hande. Thank you. Thank you, Hande, for a wonderful talk. It's really a fascinating work on the upper motor neurons, and the compounds you've described are very, very exciting. I look forward to seeing how that evolves over the next months and years. We're still a little bit behind in our schedule, and I have collected questions that have come in through the question and answer box. And so what I propose is we move on to the next talk, but we can talk either offline or at the end of the presentation about specific questions for you. So thank you so much again.
Thank you. Thank you.
Our next speaker